Hello friends, today's topic is about ALA synthase and ALA dehydratase. Now these two reactions are the first two reactions of the heme synthesis. Now each and every reaction in our body have their own significance, and particularly what is the significance of these two reactions as well as which particular disorder is associated with these two reactions that we will look in this video. So first of all, heme synthesis, first two reaction, that is ALA synthase. So here I have written that glycine plus succinyl CoA, these two are the precursor of the heme synthesis, and with the help of this ALA synthase which is present in the mitochondria, this glycine and succinyl CoA will be converted into the ALA, that is delta aminolevulinic acid. So in detail already we have discussed in a heme synthesis. Now here that PLP means pyridoxal phosphate, that is vitamin B6 is needed for the activity of this ALA synthase. Now this ALA synthesis enzyme is also of a two variety, ALA synthase 1 and 2. So further in detail that we will discuss in this video. Further, this ALA which is produced in the mitochondria that will go in the cytoplasm. With the help of that ALA dehydratase enzyme, and with the help of that enzyme, ALA will be converted into the PBG, that is called a porphobilinogen. So another name of this ALA dehydratase, that is PBG synthase. So that is another name, PBG synthase, that is porphobilinogen synthase enzyme. So sometimes it is rarely asked that another name of the ALA dehydratase. Here water molecule is removed in this dehydratase, so it is particularly name of enzyme, that is dehydratase. Now this one is X-linked type of the particularly ALA synthase 2, that is X-linked, and this ALA dehydratase, that is autosomal recessive variety of the particular enzyme. Further, this PBG, from that PBG with the help of this different enzyme, lastly heme is synthesized, but we will not discuss that. In this video we will going to discuss about ALA synthase and ALA dehydratase and disorder related to these two enzymes. So ALA synthase, that is a key enzyme of heme synthesis, so it is under the feedback control. So further, this ALA synthase is divided in a two particular category, ALA synthase 1 which is found in all of the tissue cell and most of all tissue cells containing this ALA synthase 1, and particularly in a hepatic cell. Further, this the heme is able to inhibit this ALA synthase 1, but this heme is not able to inhibit particular ALA synthase 2 which is found in a erythroid tissue. So in erythroid tissue this ALA synthase 2 is present and it is not under feedback control of the heme, and this one is particularly X-linked type of the porphyria. So if this enzyme is not working properly, and that will lead to the particularly porphyria, and that is called X-linked porphyria. So you have to remember only one porphyria, that is X-linked, that is ALA synthase 2 enzyme deficiency which is found in this erythroid tissue, and if that enzyme is defective, and that is leading to the X-linked porphyria. Here defective gene of ALA synthase, that is also sometimes responsive to the vitamin B6 or pyridoxine responsive sideroblastic anemia. Generally anemia is not observed in the case of porphyria, but vitamin B6 responsive type of the sideroblastic anemia is seen when there is a defective gene of this ALA synthase. Further, excessive production of heme intermediate causing neurological symptoms. So in this X-linked porphyria, sometimes this type of excessive intermediate formation, and that is leading to the neurological symptoms. Porphyrin precursor and porphyrin in urine and feces are normal. Here you can also understand that particularly initial step means first step of heme synthesis is affected. So ultimately this porphyrinogen and porphyrin are not excessively present in this urine and feces. So this one is particularly related to generally what happen in porphyria, that porphyrin are observed in urine and feces, but you have to remember in ALA synthase type of enzyme deficiency or X-linked porphyria, these porphyrin precursor are not found in urine and feces. Now definitive diagnosis of this ALA synthase is possible with the demonstration of mutation in the erythroid ALA synthase, because it is found, it is particularly defective in erythroid tissue. So you have to demonstrate that mutation in this erythroid, and this ALAS2 mutation is observed in this X-linked porphyria. Now further in detail of ALA dehydratase. So another name of ALA dehydratase, that is PBG synthase, that is porphobilinogen synthase, and this one is the second step of the heme synthesis. Now interesting is that ALA dehydratase, that is zinc dependent, but it is inhibited by the lead. So you have to remember two things, zinc is required and lead is inhibiting this ALA dehydratase. You don't have to you have to keep in mind that lead is not inhibiting ALA synthase, it is inhibiting ALA dehydratase. Key enzyme, that is ALA synthase, but lead is inhibiting ALA dehydratase. Further, you have to remember one more thing, that ALA synthase, that is X-linked. ALA synthase 2 that is particularly X-linked, but this ALA dehydratase which is particularly autosomal recessive variety of the disease. So autosomal recessive variety of observe with this ALA dehydratase deficiency, and the symptoms are quite similar to the acute intermittent porphyria. In this acute intermittent porphyria, particularly UPG1 synthase is defective. This ALAD, D for the DOS, you would remember it is also called a DOS porphyria. So previously X-linked porphyria, that is ALA synthase 2 deficiency. Here that DOS porphyria, that is ALA dehydratase, and it is autosomal variety type of the porphyria. Now features are quite similar as a AIP, means acute intermittent porphyria, means acute abdominal pain as well as vomiting and neuropathy is observed. So these are the common symptoms of the ALA dehydratase also. But what is the difference? Characteristic features are increased plasma ALA all over ALA level, and you increase urine ALA is observed, and decrease ALAD means ALA dehydratase activity in erythrocyte observed. So overall increased level of ALA in plasma and urine is observed, but porphyrin level as normal in urine and feces. Now prenatal diagnosis is by ALA dehydratase activity in cultured chorionic villi is possible. So you have to prenatal diagnosis of ALA dehydratase deficiency. Further, you have to differentiate this particular diagnosis by another condition like lead poisoning. So we have already said that lead is inhibiting this ALA dehydratase, but same lead poisoning is there because lead is present in so many things, and if that by mistake large dose of lead is present in the body, because lead is continuously accumulating, it will not excreted out, and there is an excessive dose of the lead, that will also lead to the inhibition of ALA dehydratase. There is not an actual deficiency of this enzyme, but this lead is present and that is inhibiting this ALA dehydratase, and that is creating picture just like a ALA dehydratase deficiency. Further, you have to separate this ALA dehydratase or DOS porphyria from the tyrosine type 1. Here fimryl acetoacetate hydrolase enzyme is defective, and here what happened due to defective enzyme activity, accumulation of succinyl acetone, which is structurally similar as a ALA, and that will create a picture just similar as a DOS porphyria or ALA dehydratase deficiency. But in this tyrosinemia type 1, there is a mental retardation as well as cabbage like order is seen. Now this tyrosinemia type 1, that is also autosomal recessive, just similar as a ALA dehydratase. But you have to remember that ALA dehydratase is not autosomal dominant, but a autosomal recessive type of porphyria is developed due to deficiency of it. Further, already we have discussed that acute intermittent porphyria. Here we are also going to discuss this ALA synthase 2, which is called X-link. So here quite summary of this important porphyria, ALA synthase 2 deficiency, that is leading to the particular X-link porphyria. Here particular cutaneous photosensitivity are more seen. ALA dehydratase, that is mostly creating just as a AIP, means acute intermittent porphyria like picture. It is also called a DOS porphyria, and this one is particularly autosomal recessive variety. UPG1 synthase, that is most common acute porphyria. So most common acute porphyria, that is called acute intermittent porphyria. These both are seen very rare in very rare type of the porphyria, and these two you have to remember it is considered a rare porphyria, and this one is the most common acute porphyria, and its incidence is around 1 in 10,000. So here acute intermittent porphyria, there is a no photosensitivity, just similar to the ALA dehydratase deficiency or DOS porphyria. There were also neuro visceral observation symptoms are observed, but particular that photosensitivity is not seen in ALA dehydratase. Further, about this different type of porphyria like particular CEP, means congenital erythropoietic porphyria, PCT, porphyria cutanea tarda, hereditary coproporphyria, porphyria vericata, and erythropoietic protoporphyria, that also we will discuss in separate video. So at present you have to remember ALA synthase 2, that is X-linked porphyria, ALAD, that is also called DOS porphyria, and that can be asked sometime in exam as well as in the Viva and MCQ question. So that's all about ALA synthase and ALA dehydratase.